right. Good morning, everyone. My name is CK, product specialist from the Instrument Division at Apical Scientific. I'll be your host for today's webinar. First of all, welcome everyone in joining us today for this webinar entitled Discover the Quantum Tx on how to count bacteria in minutes. The webinar is organized by Apical Scientific in collaboration with Logos Biosystems. Before we start, just a quick webinar housekeeping. We will begin with the webinar presentation, followed by the Q&A session in the end. So throughout the webinar presentation, your microphone will be muted to avoid background noise during the session. But if you have any questions, please feel free to submit through the chat box. We'll be answering them in the Q&A session later. So without further ado, let's welcome our today's speaker, Mr. MJ. MJ is the Apex Sales Manager from Logos Biosystems. Over to you, MJ. Thank you.
Thank you, CK. So, welcome everybody. My name is MJ. Like CK mentioned, I am the Apex Sales Manager at Align Genetics. Thank you for attending today's webinar on discovering the quantum Tx and how to count bacteria in minutes. The webinar will last about 30 minutes, and if you have any questions at the end, feel free to contact. Yes? Sorry. Feel free to contact Apical Scientific directly. They'll be glad to answer any kind of questions. First, let me share some information about my company. First of all, I want to share a brief introduction about our company. Align Genetics, formerly Logos Biosystems, was founded in 2008. We have a large network of partners worldwide with a deep focus on innovation. We have two brands, GeneScale, which is an in vitro diagnostic point-of-care solution brand, and Logos Biosystems, which is already a leader in imaging solution for life science, including digital microscopes, tissue clearing solutions, and cell counters. Today, we will specifically talk about Quantum Tx, which is our cell counter for bacteria. In Logos Bio, within the cell counter range, we have a total of 6 instruments. Each instrument has its own unique characteristics and specification. When confronted with a customer, our first question is about their cells, because depending on the type of cells, users must acquire different type of instrument. In this webinar, I will specifically be focusing on the Quantum Tx, our microbial cell counter focusing on bacteria. So from the need to accurately quantifying live lactobacillus in probiotics, heat-killed Vibrio cholera in a vaccine, sulfur oxidizing bacteria for waste treatment, or just your standard cell suspension in a laboratory, bacterial cells counting is not only fundamental, but also a significant step in all those fields. What is common to all those fields listed in the slide, which is cosmetic, environmental, food, healthcare, public research, veterinary, is the need for a standardized, accurate, fast, and ideally cost-effective solution. When you investigate existing guidelines, you notice that despite, sorry, I think there's an echo in the, thank you. So you notice that despite recommendations to validate cell counting methods for bacteria, you sometimes end up with more questions than answers. But recently, the ISO international standard was released in 2018. The USP Chapter 1223 in the European Pharmaceutical Review clearly highlights how inappropriate it is to consider the CFU, which is the colony forming unit method, as the gold standard, as well as the need for the FAST method as well. Obviously, the fact that lots of bacteria, including pathogenic bacteria, can turn into a dormant and non-cultivatable state, named viable but cultivatable VVNC, raises concerns about the standard method for the enumeration of viable bacteria. Of course, depending on your field of interest, VVNC can be more or less critical. But imagine you develop an antimicrobial treatment and you monitor the cultivability of the targeted bacteria by counting CFUs. You could obtain wrong data about the effectiveness of the treatment. Even worse, as VBNC state is not permanent, those bacteria can resuscitate in favorable conditions. Nevertheless, let's have a look at existing methods to count bacteria. Dilution method typically includes a colony counting approach. The dilution method for bacteria counting involves making a series of diluted samples from the original bacterial suspension, and then plating the diluted samples on solid growth media to enumerate the number of bacteria. This method is used to accurately determine the number of bacteria in a sample by overcoming the limitation of direct plating, where a high bacterial count can lead to overgrowth and inaccuracies in the plate count. The most common dilution method is serial dilution, where each successive dilution is made by adding a fixed volume of the previous dilution to a fixed volume of sterile liquid or a buffer. After dilution, a sample is plated on agar, incubated, and the number of colonies is counted to determine the number of bacteria per milliliters of the original sample. However, time investment is a significant drawback when assessing time-sensitive issues such as food or water contamination. Long process, it takes days, and the strength of dilution series is that it is inexpensive, but weaknesses consists of labor-intensive, time consumption, and only works for viable cells. Another common method is counting under a microscope by using a Petrov-Hauser or Levi hemocytometers. It's easy to perform, but there is a high user-to-user variability. You might know that bacteria are constantly moving and can be found on different focal planes, leading to hard-to-reproduce counts. The weaknesses of direct method is analyst-dependent, labor-intensive, time-consuming, and it only works for total cells only. Indirect method includes turbidity measurement with a spectrophotometer, dry weight, or metabolic activity measurements. Weaknesses of indirect method includes it may require high concentration of bacteria, and it must be combined with colony counting to increase accuracy. Low accuracy if the sample is contaminated, and it only works for total cells only. And lastly, flow cytometry, however, is recognized as the most reliable technique among these methods. On the other hand, it is expensive. It also requires extensive training to operate it and to analyze the data. Another significant limit of flow cytometry is the lack of analysis of clusters or chains. For example, single cells of a bacilli chain cannot be distinguished. So no solution is perfect, and the most of the time, researchers use several of these techniques in parallel. So there is a need for a rapid and accurate technologies to efficiently count bacteria. That is why we propose the QUANTOM Tx. In order to make it reliable when counting bacteria, you need to overcome some challenges. Because bacteria have certain characteristics which makes them challenging to count. The first one is diversity. We are talking about more than 30,000 formally named species that are in pure culture and for which the physiology has been investigated. and there are probably millions which remains undescribed. The second one is size. Cell size and genome size of bacteria are much smaller than regular mammalian cells. The third one is morphology. Bacteria have different shapes such as round, coti, spiral, bacilli, stars, or even rectangle, and they can form some kind of cluster or chains. Of course, bacteria are also classified as gram-positive or gram-negative depending on their wall composition. The last major challenge is that the bacteria are moving. They have a Brownian motion. On the contrary to mammalian cell lines, in a hemocytometer or slide, bacteria doesn't settle down. They are on different focal planes. So when developing our quantum TX, all of those challenges were taken into account. So you can easily guess that because of those challenges, we designed the quantum TX to not only be a cell counter, but also become a complete solution that includes the instrument along with the dedicated software. To understand how it works, let's have a look at the workflow of the quantum TX. The workflow is very straightforward, but each single step plays a critical role in accurately counting bacteria and determine the viability. So we have total of 4 steps, but we will talk about step 1 first. The first step is the sample preparation. Bacteria can be gram-positive or gram-negative, and gram-positive have a thicker layer of peptidoglycan responsible for retaining the gram stain. It means you need to use the dyes which can penetrate the wall, whatever the composition of the thickness. Therefore, we developed total of 2 kits. The first one is the Quantum Total Cell Staining Kit. It's a proprietary dye with high signal-to-noise ratio, which is much better than the acridine orange or cytone 9, which is well known if using flow cytometry. Our kit includes an enhancer and a specific buffer, a loading buffer, because we'll load the sample into the counting slides. And you want to make sure the reparation of homogeneous inside the counting chamber. The second kit is for viable staining kit. A dye is a calcine AM, derivative to the measured viability. In live cell, esterases cleave the excitose methyl group, converting the molecule to green fluorescence calcine. That being done, the first step would be to load 5 to 6 microliters of your stained sample into the dedicated counting slides. Those slides have optimized dimension and specific coating for the quantum TX. In the second step, we are going to immobilize bacteria on the same focal plane. So as you can see in the image here, you can see the cells floating around in the both top and bottom focal plane before centrifugation. However, after doing centrifugation for 10 minutes, you can see that all the cells are gathered in the same focal plane, which is at the bottom. Now we are ready to count the bacteria with the quantum TX. So quantum TX is a benchtop instrument which has a 10-inch touchscreen embodied in the instrument, and the quantum TX has highly sensitive optical design and fast scan stage and automated Z-focusing as well. And other basic specifications are listed on this slide as well. User interface has been developed to make the software intuitive for the users. The QUANTOM Tx can capture up to 10 images upon pressing count, and you can control key parameters of the protocols such as object size, roundness, and declustering level. Fluorescence objective size measures the diameter of the fluorescence signal from the nucleic acid stains, and increasing the roundness protocol will include more rounder shapes such as Kochi, and increasing the declustering level will lead to a higher sensitivity to clusters. And also higher values of detection sensitivity will detect fainter signals from the weakly stained cells or smaller sample sizes, but it can also increase the noise as well. So it is up to the customers to optimize the protocols for their cells, but normally we have tested many kinds of bacteria cells and made a default protocol. And most of the bacteria should be fine by using our default protocol. With the advanced declustering algorithm of the QUANTOM Tx, as you can see, here is the difference of results based on the protocol difference, specifically for declustering level. So from raw image to no declustering level, to low declustering level, and to high declustering level. As mentioned previously, one of the challenges to overcome when counting bacteria is morphology. However, the QUANTOM Tx is able to decluster the cells with different shapes as well. In the image, you can see that each cells do not necessarily share the same shape, but in the analyzed image, every cell is declustered to perfection. After you count, the QUANTOM Tx has a very intuitive data management user interface. After your count, you can find your counting results in the Result tab, and in this tab, you will be able to see the cell concentration, average size of the cell number, and also dilution factor. You can also click the Save and Print section to review and save your data through the SSD drive. In the saved file, you can include analyzed images, raw images, PDF reports, and also CSV files in just the one click. So you have seen the QUANTOM Tx workflow is simple but optimized to efficiently determining bacteria concentration and viability. And this can be done in less than 45 minutes in total. And in the first stage, it says 1 to 30 minutes because the incubation of sample at 37 degrees for a minimum 20 minutes is required for counting bacteria. So a good number of bacteria have been validated. Here are some examples. More data can also be found on our website. Of course, we didn't have chance to test the all 30,000 bacteria strains which have been characterized over time, but our solution is flexible enough to work with gram-positive and minus cells as well as the VVNC states. So I will now share with you some applications based on the published papers. First application is in antibiotics. Antibiotic resistance poses a huge threat to the effective treatment of bacterial infections. Researchers are attempting to repurpose pre-developed drugs that are known to be safe. And these authors also noticed an increased cell size and decreased motility due to cyclopyrax. And the second application is in food safety. Data was obtained from records collected by trained research officers at Dairy Services Zimbabwe from 12 large-scale and 16 small-scale randomly selected milk processors. Understanding how structural condition of processing facilities and food safety practices affect E. coli, and coliform's presence in cultured buttermilk, which is one of the most popular products of milk, enables processors and regulatory agencies to put in corrective actions using a cell counter to reduce microbial contamination of cultured buttermilk during processing, especially by small-scale milk processors. It helps to reduce the risk of consumption of contaminated buttermilk. And shelf life of the cultured buttermilk is also improved as well. Third application is in probiotics. Lab strains could improve the quality of fermented foods such as yogurt, soy sauce, sauerkraut, fish or meat products or pickles, and etc. by improving the taste and health-promoting benefits of the product as well. The fourth application is in cosmetics. We used the QUANTOM Tx to compare urine samples found to be positive for E. coli via cell culturing with UTI negative samples. And the patients with the positive urine culture had significantly higher cell count results using the microbial cell counter. And the fifth application is in immunology. Breathing allows multitude of airborne microbes to access the lungs, and discrimination between live and dead bacteria from lungs has been suggested to understand proper control mechanisms, ensuring the differentiation between threatening and harmless encounters. Altogether, our result shows that the PCLS adapt the extent of cytokine expression to the viability status of P. aeruginosa by specifically internalizing live bacteria. And this is, I think I mentioned this previously in the cosmetic field, but the sixth application is in potential clinical application. And the QUANTOM Tx was used to compare urine samples, and patients with positive urine culture had significantly higher cell count results using the microbial cell counter. And there is also a great deal of interest in finding newer, faster, and more reliable methods for diagnosing UTI as compared to the standard urine culture. And this is the last application I will be sharing with you today. Also, here is some of the key features of the QUANTOM Tx. It is advanced image-based cell counter, and it has new fluorescence staining dye for total cells and live cells. It is compatible with gram-positive and negative and VVNC state cells, and it has efficient cell immobilization with our QUANTOM Tx centrifuge. Has highly sensitive optical design, has large counting volumes, which has 10 fields, fast scan stage and automated Z-focusing, and fast and accurate cell counting algorithm as well. And bacteria-specific declustering algorithm has been implemented into the instrument as well. And the instrument has 10-inch touch screen intuitive user interface, and onboard storage, and multiple export options as well. We have a high quality product and we work with the same standard for customer support as well. Here are some of our customers kindly wrote the reviews on the Quantum Tx. You can find it on our website or other platforms like Select Science as well. Okay, that is the end of the presentation. Thank you so much for attending this webinar, and I hope the content was interesting and relevant for you.